be microscopic images, and we're going to go over again what you need to know. You can follow along in your lab objective sheet. We're going to start here in the oral cavity, looking at a section through the tongue. And you're looking here at the tongue, extensions on the surface of the tongue. There are three major ways we do it. Which one is this? Circumvallate patella. These are the largest and the least numerous of the three general types of patella. And they're covered with the same type of epithelium that lines most of the oral cavity. And what kind of epithelium is that? What are we dealing with? We're only dealing with two types of epithelium in the GI tract. What is this? Non-keratinized stratified squamous epithelium, yes. So that's what we see here. Multiple layers of flattened cells. And again, you can tell by looking at the nuclei. You really can't see the cell boundary. You can see the nuclei are flattened. There are obviously multiple layers. It is stratified. So it's stratified squamous epithelium. Again, it's non-keratinized, unlike the epidermis, the skin. Also embedded in the epithelium, we see some important structures. These structures right here, what are they? Those are the taste buds, which give us the receptors for our basic taste sensations: sweet, sour, salt, bitter. Again, the subtleties of taste discrimination, which humans are capable, has more to do with your sense of olfactory smell than with these receptors. And of course, the olfactory receptors are located in the nasal cavity. So when you congest, you have a lot of mucus production, allergy, or upper respiratory tract infection. Food tastes rather bland because you can't smell it. As a result, you're basically limited to the sensations as a result of these receptors, the taste buds. So, circumvallate papillae, the largest of papillae, also the least numerous, and they have taste buds embedded in the epithelium. Also in the tongue, you'll see the other two types of papillae, but not necessarily on the same slide. And in this one, if we go out to the edge, you can see a very good example of another type of papilla. What is this? That's the filiform. Filiform papillae, you can see, are spine-like. And actually, they're keratinized, stratified squamous epithelium, and no taste buds associated with it. What's the function of these filiform papillae? They give the tongue a rougher surface. They increase the grip of the tongue. They don't move the food. The tongue does that. They allow the tongue to do a better job of it by giving it a better grip. So they increase the grip of the tongue by giving it a rough surface. Okay, and some animals will actually groom themselves by licking their fur, basically they're combing debris out, and they have a lot of these papillae, these sharp papillae. So these are the filiform, we just call the circumvallate. These are the smallest and the most numerous. But again, no taste buds associated with them. Also in the tongue slide, you can see some other important structures. You see these dark purple cells here, what are they? These are serous cells. Remember, the salivary glands, it's a combination of two general types of cells, serous cells and mucous cells. And usually the darker stained ones are the serous cells. And they produce what kind of secretions? Thin, watery secretions high in what? Amylase. And that's a digestive enzyme. What does it do? It breaks down complex carbohydrates like starch, in this case into maltose, which is disaccharide, a double sugar. So carbohydrate digestion actually begins in the mouth because of the salivary amylase that we see in secretions of these serous cells. You're also, of course, in the tongue. See this? What is this stuff? That is skeletal muscle. That's why I'm going to accomplish so many movements with the tongue. Of course, skeletal muscle is the only one that's under conscious control. Smooth muscle and cardiac muscle is involuntary. It's not under conscious control. You can't decide when to contract and relax. Okay? So again, this slide is the monkey tongue taste buds. This next slide is labeled lingual glands, and it shows again a section through the tongue, and then also you're going to see the same type of cells we saw before, serous cells and also some mucous cells. And here again we see the surface of the tongue. Again, it's non-keratinized stratified squamous, as we saw in the previous slide. What do we have here? A filiform papilla again. And now we see the third type of papilla, these rectangular ones here. Again, they have taste buds embedded in the epithelium. What are these? These are the fungiform. Fungiform are intermediate in size and abundance, which means they're smaller than the circumvallate but larger than the filiform. They're more numerous than the circumvallate but less numerous than the filiform. But again, both circumvallate and these fungiform papillae have taste buds embedded in our epithelium. And then again, if we look deep here in the connective tissue, we can see, again, these purple stained cells. What are they? These are the serous cells again. What kind of secretions? Thin watery secretions, high in salivary amylase. Okay. Here's the other type of cell, though. Now, what kind of cell is this? What are these cells? These are the mucus cells. Okay, they're usually lighter stained. What kind of secretion they produce? They got a lot of mucus. Okay, mucin. All right. So this is a thick viscous secretion, low in enzyme. So our salivary glands have a combination of both types of cells. In this case, it's a lingual and sublingual glands, which are associated with the tongue and under the tongue. Now, what's the ratio here of serous to mucus cells? One more abundant than the other in these glands? More mucus cells. It's actually more mucus cells, which means the secretions of the lingual sublingual glands are going to be low in enzyme and more thicker and more viscous. Okay? And again, you can see skeletal muscle as in the previous slide. Okay? So you should be able to recognize these types of cells, know the type of secretion, like I say. The salivary glands have combinations of these. In the case of lingual and sublingual, it's more mucus glands. Any questions? All right, let's look now at the submandibular salivary gland. And again, this has both types of cells, mucus and serous cells. We're going to see some other things as well. Again, the darker stained cells are going to be the serous cells, and the lighter stained ones will be the mucus cells. So these dark purple ones here are serous. And the lighter ones that are kind of gray, like we see here, okay, these are the mucus cells. We've also got a lot of ducts here. And these are salivary gland ducts. And how can we tell the difference between the salivary gland ducts and blood vessels, which we also see in these tissues? So here again, mucus cells, kind of gray, and then the darker stained serous cells. So we should be able to distinguish between these two. And what is this? Salivary gland duct. How do we know it's not a blood vessel? Look at the wall. What kind of epithelium do we have here? Simple columnar, as evidenced by the circular, okay, excuse me, simple cuboid, as evidenced by the circular or spherical nuclei. Single layer. So the small salivary gland ducts are usually a simple cuboidal epithelium. The large ones you're going to see are often stratified. But all blood vessels, from the smallest to the largest, both in veins and arteries, line with what kind of epithelium? What distinguishes blood vessels from these ducts? Simple squamous, okay? Remember, that's the endothelium, the tunica intima or tunica interna. Simple squamous epithelium, a little bit of loose connective tissue. That's all there is. Right from the capillaries, right up to the largest veins and arteries. So blood vessels are always going to have simple squamous lining, okay? And these ducts, the small ones, usually simple cuboidal. And the larger salivary gland ducts, usually stratified. We're going to see a lot of them in here. Now here's a blood vessel. It's hard to tell, but you can see it's got flattened nuclei here, which is the tunica intima. simple squamous lining. Okay, here again we've got a couple tubes. We got one right here. And what is this? What is it? It's a blood vein, okay? Again, look at the nuclei. Flattened, single layer. This is a vessel, and it's a blood vessel, and it is a vein. How do we know it's not an artery? You can see it's very long, very large, relatively thin-walled, very large lumen, and usually collapsed or partially collapsed when there's no blood in it. Remember, veins tend to be larger than the corresponding artery, and since they carry blood at much lower pressure, a much thinner, weaker wall. And that's why they usually don't hold their shape in these tissue sections when there's no blood in it. All right, right next to it, you can see there's another tube. This one you can see looks a little different in the wall. It's not simple squamous anymore. It's actually stratified. And if you look at the shape of the nuclei, you can see they are in fact oval, which indicates what kind of cells? Columnar. So this would be a stratified columnar epithelium, which is relatively rare in the body, but we do see it here in the large salivary gland ducts of the submandibular salivary gland. So in case you can see it. Again, look at the nuclei. Again, it keeps working on the focus here, but you can see they are more than one layer of nuclei. And the nuclei are basically oval in shape, as you can see. So stratified columnar epithelium, typical of the larger salivary gland ducts. You can especially see it here in the submandibular salivary gland. So again, you should be able to pick out the serous cells, the mucous cells, tell the difference between the blood vessels lined with simple squamous, all of them, and then these ducts. Simple cuboidal epithelium for the small salivary gland ducts, and a stratified, usually a stratified columnar in the larger salivary gland ducts. Here we can see some small blood vessels here. And again, you got the simple squamous lining basically. You can see some red blood cells in